Alright, ladies and gents, welcome back. We're going to keep discussing the steps of your scientific method. So yesterday we talked about your two major beginning steps, right? So really just narrowing down what that question is, stating what your problem is that you're ultimately going to test, and then doing that step two that we don't want to forget, that collecting additional information that's going to ultimately help refine that question. And really that information then is also going to get used in this first step we're going to talk about today, and that's forming a good hypothesis. So most students, when they're initially taught what a hypothesis is, you're told that it's an educated guess. I really hate that word guess, and in fact we're going to cross it off in your notes and we're going to change it to an educated prediction. Why don't I like that word guess? Well, because it sort of implies that you're literally guessing. You're taking a shot in the dark as to what you think is going to happen. And realistically, a good hypothesis shouldn't be a guess. You're making a prediction about what's going to happen at the conclusion of your experiment, but it's an educated prediction, meaning that you've done that background research in step two. You've seen what others have done in either the same type of experiment or similar situations to what you're researching. And so a good hypothesis should be based on some of that previous research you're already doing before you do the experiment. So no longer will we use the word educated guess. I see it on a test, I see it in an answer, and I promise you I will take points off. Tell me that it's an educated prediction because you've done some background research already. So to be a truly complete hypothesis, I'm not just stating what I think is going to happen, but I'm telling my reader, or I'm telling myself, or I'm telling my research team why I think that this is the prediction that I'm expecting to see, the outcome I'm expecting to see, and it's because I have done that previous research, okay? So that's something I'm going to nitpick on. It is not an educated guess, it's an educated prediction because you've done step two. You've done that extra research or those additional observations. So if we were to define what a hypothesis is, remember, we're not using guess anymore, prediction, but it's a tentative explanation. And that explanation is going to account for a set of facts that you've already researched back in step two that we can then test further using some sort of controlled experiment. So, in order to be a truly reliable, functional hypothesis, it's got to meet really two criteria. One, I have to design my hypothesis, I have to write it in a way that I can actually test it. It needs to be testable. So I need to be able to design a controlled experiment around the way I've written my hypothesis that is actually going to get me data that I can collect, that I can analyze, and that I can draw conclusions from. The second big piece is that a hypothesis has to be falsifiable. I have to be able to write a hypothesis in a way that has the potential to come out unsupported. It is possible to write a hypothesis that no matter what data I collect, I will always end up supporting it. That's not a good hypothesis. You've already predetermined what your outcome is going to be before you have ever gotten there. So you want to make sure that I can test it, so it's in and of itself a topic I can test and design an experiment around, and then two, that I have done it in a way that can either be supported or has the potential to not be supported. So the initial steps in making a good hypothesis are to make some predictions, to forecast or guess the outcome, predict the outcome of a situation beforehand. So I'm guessing most of you have probably been taught to write a hypothesis as an if-then statement. So if I do this, then this is what I think is going to happen, right? Truly, if all I do is stop at an if-then statement, I've really just written a prediction. So let me just give you some examples, right? So let's go back to the watery, soapy water experiment with the plant, right? The question that I was asking is, does soapy water kill my plants? So if I'm writing a prediction for that question, I might say, well, if I water plants with soap water, then the plants will die, right? So I'm saying if, and then here's the variable that I'm going to change to test my hypothesis. So the soapy water. Then I state what I think is going to happen. Well, in this case, then the plants will die, right? Or if I use the goldfish and food example, if, here's the variable I'm going to test, so feed the fish different foods, then here's what I think is going to happen. Then they will eat more of this particular Miller brand flakes, okay? This truly is just a prediction, however. Okay, in order to make a good hypothesis, I want to take it one step further. So there's my plants being exposed to the soap, they're going to die, right? If I do this, then this will happen. There's the prediction. But a good hypothesis is going to include one more piece. So if I expose the soap to the plant will die because, and here's where step 2 comes in, here's where your rationale gets added in, because the soap breaks down the cell wall, which is needed for the plant's protection, right? So, prediction, fish prefer Miller brand flakes, right? If I feed them different foods, then they will choose the Miller brand flakes. But a hypothesis then includes a rationale. So fish prefer the Miller brand flakes because this has a more balanced nutrient content. Okay, so really good maybe side note for yourself is that if I'm writing a truly good hypothesis, it's if I do this, then this will happen because here's the rationale that I have for making that prediction in the first place based on the research that I did back in step 2, right? So a really good hypothesis, and I want to see this pattern when you're writing these in test questions, reading guide questions on your labs themselves, if, then, because, right? Once I've got that if, then, because in place, I can actually design a really good experiment because I've already designed a really good hypothesis. So, designing your experiment then is a way to ultimately test that hypothesis. So I want to make sure first off that I'm creating a controlled experiment, or at least as controlled as I can get it. So what do I mean by a controlled experiment? In a controlled experiment, I am testing only one variable at a time. Every other variable that's going on in that experiment should be identical to each trial, to each group that I'm doing, to ensure that my experiment is fair and that I can actually have some reliability and confidence in the results that I'm getting, and that those results are based on the manipulation that I'm purposefully doing and not because something else is going on in the background that I'm not controlling for. Okay. Now, realistically, can every experiment be truly controlled? Of course not. And in biology in particular, having that truly controlled experiment does get tough with living systems. But the closer that we can get to a controlled experiment, the better off we are. Alright, so that's always the ultimate goal. So when we design controlled experiments, we've got a couple of things that we need to make sure are set up in place properly. First off, the hypothesis in and of itself, but then designing an experiment where I've got two groups to work with. So one being your control group, this is the group where all the conditions remain absolutely identical, except they're the ones who aren't going to actually get the variable you're testing. Your experimental group, on the other hand, absolutely identical, or at least as closely identical as possible to your control group, except here's now where I'm going to change one thing, and that thing is ultimately the variable that I'm changing because it's the question that I'm asking. It's what I'm testing for. So if we think about a human drug trial, right? We're testing some new medication. Let's just say it's a medication for high blood pressure. I'm going to split out my test subjects, my patients, into two groups. One set of individuals is going to get the placebo, all right, or a sugar pill. That's your control group. I'm giving them everything that's identical, all the way down to giving them some sort of pill. But my experimental group is the one that actually gets the medication itself, right? So both groups might be similar age brackets, might be obviously having similar condition of high blood pressure. They might be similar, maybe they're the same gender, maybe they have similar diets, whatever the case might be. I'm trying to control all of the variables between my control group individuals and my experimental patients so that they're identical, or at least as close to identical as possibly feasible, all the way down to actually giving them both medication that they think they're getting the real thing, right? My experimental group is actually truly getting the medication that I'm testing. My control group is still getting a pill thinking they're getting the medication. And truly, why do we do that? Because that way I can control and compare the results that I get in my experimental group to my control group. We talk about this as a placebo effect. So, do the patients just think they're getting better because they're getting this pill? Or are they really getting better because of the medication? So our ultimate goal here by setting up these two groups is to see what we would say is a statistically significant difference in my experimental group compared to my control group. So that whole point of that control group is a comparison. If I see 50% of my experimental patients get better, and I see 45% of my control group patients get better, it's the placebo effect. It's just because they think they're getting the medication, and it's not truly the medication itself that's helping them. On the flip side, though, if I see 50% of my experimental group get better, and only 10% of my control group gets better, now I know it's probably the medication that I'm giving them that is really making the true effect in lowering their blood pressure, for example. So that control group gets set up as a comparison. But only one thing changes. Only the actual medication in this particular example is what changes. Everything else I want to try to keep as identical as is possible. Once I've established what those groups are, I can then establish the variables themselves. So a variable in general is just any factor in an experiment that could ultimately affect the results, and so I either want to make sure that I'm keeping it consistent between those two groups, or I'm running separate experiments to test those variables separately. So we break them down into a couple of different categories. Your independent variable, this is the one that you are purposefully changing. This is your manipulated variable. So in the case of my drug trial, do they get the placebo or do they get the real drug? That is my independent variable. That is what I'm purposefully changing between my control group and my experimental group. The dependent variable, on the other hand, is the one that responds to the change. It's your responding variable. Or, side note here, I tell kids to think about it as your data. It's what you're collecting to see whether or not your manipulation of that independent variable really had any effect. So I'm taking a look back at that drug trial and I want to know how many patients see a decrease in their blood pressure. That is my dependent variable. So 50% of my experimental group saw a decrease, 10% of my control group saw a decrease. That number, that observation, that data is your dependent variable. Everything else I want to control for. I want to keep the same. So going back to those two drug trial groups, they should have the same disease, right? In this case, we're talking about high blood pressure. They should probably be within a similar age range. High blood pressure in a 5-year-old is going to work and function and look a lot more different to high blood pressure in a 50-year-old. So maybe we are keeping it to, say, 40 to 60. All right, so adults. We may find that gender plays a role, so we want to keep everybody in both groups male. We find that activity level plays a role, so we want to make sure that every patient in both groups is exercising 30 minutes 3 times a week. Food may play a role, and so we try to keep their diet as similar as possible. Those are all controlled variables that we believe could have an impact on our high blood pressure, and so we want to control for that. We want to keep them the same so we know that it really is just the medication that's changing their blood pressure and not some other extraneous variable. Those controlled variables are the hardest ones to keep in check, especially in biological systems, and even more so in living organisms, because there's only so much control we can have outside of the actual lab itself. But the more we can control, the better, and the more reliable our results are. When we come across things that we can't control for, we find out after the fact, or we just know there's no way to keep those under wraps. Those are then things that we have to make sure that we report, and we'll talk about those things as sources of error a little later. Going back to just some graphing, so when I'm producing graphs of my data ultimately at the end of this and part of that analysis piece, something to keep in mind is that there really is a truly best practice way to set up those graphs in terms of the variables in the data that you have. So your independent variable will always go on your x-axis, and your dependent will always go on your y-axis. So how do you remember? Just a quick little acronym. So DryMix seems to work for a lot of kids. So dry, your dependent or your responding variable always goes on your y-axis. Alright, and your manipulated or independent variable, the mix part, goes on the x. Now, I will warn you that this particular acronym really works well if you've got a single variable that you're testing, single set of data that you're working with. If you start seeing multiple line graphs or multiple plots on a single graph, then you've got to be careful looking at your keys. A lot of times you'll still see your dependent variable along your x-axis, but the independent variable is probably the different lines that they're plotting. Alright, once again, maybe not a bad side note, right? So dependent variable probably is still on your y-axis, but your independent variable is probably in the key itself, and it's the different lines that they're plotting.